Hey everybody, it's Dr. Gilchrist, and obviously we have day 2 of our snow and extreme cold days. Um, I am pretty sure we will be in class on Friday because it is above freezing. For today, I wanted to continue our Lecture 14 slides, and I'm going to try to finish these up today. Now, I did mention on Monday that this will probably have one of the tougher things to talk about in this class. So while I'm not happy about not physically being in class, I think this is a case where it may have worked out for the best, because for this tricky part that we will be talking about today, you now have an opportunity to go back and listen to this lecture whenever you want. You have an opportunity to go back and listen to it more than once just to make sure you feel like you really got it. So the last time we were here, we were talking about the major components of the eye. And as I kind of mentioned, it's a very common metaphor to liken the eye to a camera. So you can see here that light will basically enter through the cornea into the pupil and will strike the lens. The lens is going to refract or bend that light onto the retina of the eye. Now remember that our centermost vision is in a region called the fovea. The fovea has the greatest amount of cone photoreceptors, which, as I've already talked about, work especially well for processing color. And we'll talk more about the photoreceptors and what they do in a couple of slides. But getting back to this metaphor as the eye functioning as a camera. So cameras have lenses, and we can change the quality of a photograph by changing the nature of the lens. Now, in our eye, our lens can actually change its shape to focus on objects that are far away or close up. Our lens's ability to change its shape, a process known as accommodation, actually gets worse with age. The lens becomes more brittle, and as a result, many people will require glasses to be able to focus up close. So changing the lens shape can change the quality of the images. It can also change our focus. The amount of light is going to be controlled by a camera's aperture, but our iris is what is allowing light to enter the camera. So in very dark situations, the iris is basically going to make the pupil look very large because it's looking for any photon of light that it can. In very bright conditions where there might actually be too many photons, the lens will the iris will restrict and make the pupil very small because we don't need all of the light that is available. And then finally, the light is going to strike the retina, and that's going to enable us to see images, especially as we talk about the makeup of the retina. The rods and cones in the very back layer, the bipolar cells, and then the ganglion cells right here, whose axons form the optic nerve that will leave the eye. So light is basically hitting the retina. If we're talking about a camera, that will be either film or photographic paper. So now let's go ahead and talk a little bit more about the makeup of our retina. So we're starting at the very back with our photoreceptor layer, our bipolar cell layer, and finishing up at our ganglion cell layer. Now there are 3 different layers of the retina, and one thing that I really want to impress upon you is that all of these are neurons. So photoreceptors are specialized sensory neurons. Bipolar cells are functioning as neurons, as are ganglion cells. Now you will also notice that embedded in these different cellular layers are what are known as horizontal cells, this cell coded in blue, and amacrine cells. So horizontal cells are basically bridging Photoreceptors and bipolar cells. Amacrine cells are bridging bipolar cells and ganglion cells. Now, both of these are going to be inhibitory interneurons, and both of them are going to play a really important role in basically sharpening contrast. So something that I talk about in my sensation and perception class when it comes to vision is that really, for us to be able to notice the edges of objects, we need to be able to understand contrast between areas of light and areas of dark. And to be honest, the world, from a visual sense, is basically made up of fuzzy stripes, edges, and contours. And this is what's going to allow us to perceive contrast in visual scenes. And we'll talk more about the horizontal and the amacrine cells and how they contribute to this understanding of contrast later on. Now, one thing that is really important, and this might be a test question, is that only the ganglion cells are going to fire action potentials. So you can see I've kind of bolded it here. Only our ganglion cells form action potentials. Both the photoreceptors and bipolar cells, while they are neurons, they have what are called graded. So remember we talked about those graded potentials where you get more depolarization or hyperpolarization based on stimulus strength. Well, that's going to happen here with our photoreceptors and our bipolar cells. They get variable amounts of neurotransmitter release. In this particular case, the neurotransmitter is going to be glutamate. So glutamate for the visual system is going to be really important for our being able to see. Now, this is kind of different from the neurons that we've talked about in class already, where we actually fire action potentials and they're critical for neurotransmitter release. But in the retina, all cells are going to release glutamate in proportion to the strength of that graded potential. So the stronger the stimulus, the greater glutamate release we're going to get. Now, the reason that they work this way is because we have variable amounts of light in visual space. Visual processing is really complex, and this is one of the ways that we can actually code for the different intensities of light that we are seeing out in the world and sending that message to the brain. Now, each particular type of cell in these 3 layers of retina is going to be responsible for a different phase of that sensory transduction. So as a reminder, transduction is basically the process of taking physical stimulus energy. In the case of vision, we're talking about light, and basically turning that into an electrical signal that the brain can actually use. So let's start by talking about our rods and cones, our photoreceptors. So as we kind of saw in that exercise that I showed you on Monday, cones are largely going to be in the fovea, and rods are going to tend to be more in our peripheral vision. So cones are going to be responsive to bright light. They are responsible for our color vision, and they are allowing us to see fine detail because they are in that foveal region, which is where our vision tends to be the clearest and have the best acuity. Now we have rods. Rods are going to be more numerous than cones. So we actually have more rods than we do cones. And they're going to be working better in dim light. They're going to be also potentially used for objects that are in motion, and they are largely used for our night vision. Now, if you take a look at the actual structure of rods and cones, you can see where they get their name. The outer segment of a cone looks like a cone. The outer segment of a rod looks like a rod. So both of these have inner and outer segments. So the inner segment of each of these photoreceptors is where the nucleus, the cell body, ultimately is located. You can see that we have our synaptic terminals, which are going to release glutamate onto onto our bipolar cells, but it's the outer segment that contains photopigment. So let's kind of break down the difference between our rods and cones. So we have about 120 million rods in each retina, and as I kind of said, they tend to exist more in the periphery. They work best in low light. They do not code for color. They code for black and white. Now, in particular, one of the things that I mentioned is that the outer segment contains a photopigment, which is also referred to as an opsin. Now, for rods, all rods carry the same type of photopigment or opsin. It is called rhodopsin, or literally translated into the opsin for rods. So the opsin for rods, the photopigment for rods, is rhodopsin. And then something else that we will talk about a little bit later is something called convergence. So if you go back and take a look, you can kind of see that a couple of different photoreceptors are converging onto one bipolar cell. This is convergence. So basically, how many rods and cones are converging onto one bipolar cell? And in the case of rods, rods have what are called high convergence. Many of them will converge upon the same bipolar neuron. Now let's talk about cones. So cones are fewer than rods. They have about 6 million in each retina. They tend to be in the fovea, and cones generally are not present in the periphery. And this is part of the reason why we have a really hard time noticing color in our periphery. We will often notice that something is moving in our periphery before we actually notice what the color is. Cones work best in bright light, and they enable us to see color vision. Now, whereas rods only have one type of photopigment, there are different types of cones. There are 3 different types of cones. So we have one type of cone that codes best for short wavelengths, which corresponds to the colors blue or purple. We have one particular type of cone that codes for medium wavelengths, which corresponds to the colors green and yellow. And then we have a type of cone, the 3rd type of cone, that codes best for long wavelengths, which corresponds to the color red. Each of those 3 cones will have its own opsin, but I am not going to name them for you here. And whereas rods have high convergence, where multiple rods will converge on the same bipolar neuron, cones have low convergence. Very few cones will converge on the same bipolar cell. So now let's talk, and I'm going to spend a little bit more time on this slide. So phototransduction is basically what's going to initially happen in both the rods and the cones. So phototransduction is basically the process through which our photoreceptors are going to actually convert physical stimulus energy of light into an electrical signal. So this is a cross section of the lamella. So one lamella, kind of one slice of the outer segment of the rod. This will work similarly for cones, but it's a little bit more complicated because each cone of those 3 types of cones has its own opsin. But please be rest assured that this basically will work in a very similar way for cones. Now, photoreceptors are not like other neurons. So basically, let's talk about what happens in the dark, and then we'll talk about what happens in the light. So this top portion right here that I'm circling with my cursor is what is happening in the dark. So there are leaky sodium channels and there are leaky calcium ion channels that are open. You can actually see those channels right here. So here we have a leaky channel, and remember that a leaky channel is always going to be open. Now, what is holding these leaky channels open is a little molecule of cyclic GMP. This is a nucleotide, and this is going to be different from other types of neurons. This is not something that other neurons do. This is only something that photoreceptors do. In any other neuron that we look at, you've probably noticed I didn't really talk that much about leaky sodium channels, and I didn't talk about leaky calcium channels. So basically, in the dark, cyclic GMP is going to hold these leaky channels open. And so the positive ions or cations, so positively charged ions like sodium and calcium, are going to flow into the cell, as you see here, and they're basically going to basically create depolarization in the dark. Now, because calcium is involved here, calcium will cause vesicles to dock at the synapse and release neurotransmitters. So in particular, the neurotransmitter that we are talking about here is glutamate. So in the dark, these leaky channels will stay open. Sodium and calcium will flow inside of the cell and depolarize it. And because calcium is entering the cell, we get more glutamate release in the dark. So in the dark, our rods and cones are depolarized. So sometimes we're going to call this dark current. So basically, anytime that we are in the dark, we are getting a constant flow of glutamate from our photoreceptors. But we're talking about what happens in the dark. What happens in the light? Now, this is going to work similarly for cones, but we're talking about what happens with rods here. So first, what's going to happen? You can see that molecules of photopigment are embedded throughout this lamella of the outer segment. Light is basically going to strike that rhodopsin, and that's going to break them apart. And the rhodopsin molecule, as you can see in this figure, will split. And the retinal portion, which is made up of a lipid, is basically going to activate a G protein here called transducin. Transducin is going to activate an enzyme called phosphodiesterase. Most of the time, if it ends in the word ase, it's probably an enzyme. So phosphodiesterase is basically going to break apart that little cyclic GMP molecule that has been holding the leaky channel open. So when the cyclic GMP is destroyed, that leaky channel closes, and ions of sodium and calcium can no longer flow into the cell. As a result, the cell begins to hyperpolarize and less glutamate is going to be released. Now, as I mentioned, this is going to happen in the cone too, but because each of the three cones has its own opsin protein, we're going to see some slight differences because they are responsive to very specific wavelengths of light to convey information about color. Now, we'll talk more about cones later, but this is the basic phototransduction process. Rods and cones are going to be depolarized in the dark, and they're going to be hyperpolarized in the light. So now let's talk about what happens after phototransduction, particularly when light has hit rods and cones and they become hyperpolarized. So just as a reminder, again, we have talked about this first layer. So photoreceptors are depolarized in the dark and they're hyperpolarized in the light. What happens next is going to be dependent on the particular bipolar cell that is directly connected to a photoreceptor. So we are about to hit probably the trickiest part of this lecture. So I would encourage you to, if you're struggling with this, listen to it again, and feel free to email me or talk with me after class on Friday in case you have any questions. So what happens next is going to depend on the bipolar cell and the ganglion cell that we're dealing with, because there are different types of bipolar cells and there's different types of ganglion cells. So we're going to start by talking about on bipolar cells. So on bipolar cells are called on cells because they are depolarized when light hits them. So in general, on bipolar cells are depolarized in the light and they're hyperpolarized in the dark. So let's start by talking about what happens in the dark, and then we'll talk about what happens in the light. And you can see here, for your convenience, I have included a summary statement in bold. So in the dark, as we talked about, the photoreceptor is going to be depolarized. As a result, it's going to release glutamate onto the bipolar cell. And this is going to have a very strange effect and unexpected effect of hyperpolarizing it. So again, glutamate release from a photoreceptor is going to hyperpolarize a bipolar cell. And this is because bipolar cells have critical potassium channels. And when glutamate binds to those potassium channels, those channels are going to open and potassium is going to leave the cell, and thus in the dark, potassium will leave the cell and the bipolar cell will hyperpolarize. So that's what's going on in the dark. The photoreceptor releases glutamate onto the bipolar cell, which binds to a potassium channel that causes the channels to open or stay open. Potassium will leave the cell, and remember, potassium carries a positive charge, so when it leaves, it hyperpolarizes the cell. Now let's talk about what is going on in the light. In the light, the photoreceptor is hyperpolarized, and glutamate will stop being released. This is going to have the effect of depolarizing our bipolar cells, our on cells, because those glutamate receptors are no longer binding the potassium channels, and thus potassium can't leave. And so because potassium can no longer leave, that is going to depolarize our on bipolar cell. The on cell will then release glutamate to an on ganglion cell, and action potentials will be sent to the brain signaling that light has increased. So on bipolar cells are depolarized in the light and they're hyperpolarized in the dark. Again, in the dark, glutamate is going to cause potassium channels to stay open. As a result, potassium will leave the cell, and in the dark, the on cell will hyperpolarize. But in the light, glutamate stops being released, potassium channels close, and potassium can no longer leave the cell, effectively depolarizing it in the light. Then we have off bipolar cells. Off bipolar cells work like our rods and cones do. They are depolarized in the dark and they're hyperpolarized in the light. So these are going to be inhibited when light hits them. So let's talk about what happens in the dark. In the dark, our photoreceptor is depolarized. Remember, we've got that dark current going. We've got those leaky sodium and calcium channels staying open. So the photoreceptor will be depolarized in the dark, and it'll release glutamate onto the bipolar cell. And this will have the effect in the dark of depolarizing it. Now, the critical ion channel for off cells is sodium. So off bipolar cells have sodium channels which can be opened when glutamate binds to them. So when glutamate is released onto the photoreceptor, from the photoreceptor to the bipolar cell, this is going to bind to the sodium channel. It's going to open the sodium channel, and sodium is going to rush inside of the bipolar cell, and that's going to depolarize it in the dark. So sodium will enter the cell and depolarize it in the dark. Let's talk about what happens with light. Now, photoreceptors are going to be hyperpolarized in the light. Glutamate will stop being released, and this will hyperpolarize the off cell because those glutamate receptors won't be able to cause the sodium channels to open, and sodium will no longer be entering the cell. So glutamate stops being released from the photoreceptors. That causes the sodium channels to close. And because of that, sodium can't enter the cell, and that effectively hyperpolarizes the cell in the light. So the off bipolar cell will release less glutamate onto the off ganglion cell, and the off ganglion cell will release fewer action potentials to the brain, signaling that light has increased on these particular cells. And remember, ganglion cells are the only cells in the retina that can fire action potentials. So again, off bipolar cells are depolarized in the dark, and they are hyperpolarized in the light. In the dark, glutamate release from the photoreceptors will cause the sodium channels to stay open, allowing sodium to flow inside of the cell and depolarize the off cell. In the light, the photoreceptors hyperpolarize and reduce the glutamate release. And because of that, that causes the sodium channels to close on the bipolar cell. Sodium will stop entering the cell, and that will effectively hyperpolarize the off cell. So in essence, these properties allow for one population of bipolar cells that gets excited by light, the on cells, and another population, the off cells, that get inhibited by it, even though all of our photoreceptors have the same response to light. All photoreceptors depolarize in the dark and hyperpolarize in the light. So we're getting more complex with each layer of the retina. And this complexity is going to be very important and necessary for detecting variability in light, including things like color, contrast, edges, etc. So again, just a review slide for you. So our on and off bipolar cells. So on cells in the dark, our photoreceptors are going to release glutamate. That's going to open our potassium channels. We're going to get an efflux of potassium, meaning that potassium leaves the cell. This will have the effect of being hyperpolarized, and it will reduce glutamate release. In the light, those photoreceptors release less glutamate, and because of that, potassium can't leave. As a result, it becomes depolarized and increases glutamate release onto ganglion cells, which will increase their action potentials. Our off cells in the dark, our photoreceptors release more glutamate in the dark. This will have the effect of opening sodium channels in which will cause a sodium influx. Thus, we get a depolarization of off cells in the dark with enhanced glutamate release onto ganglion cells. In the light, sodium can no longer enter the cell because there's less glutamate release from photoreceptors and those channels can't stay open. As a result, we get a reduction in glutamate release onto ganglion cells, and thus we get a reduction of action potentials in off ganglion cells. So if on and off cells are near each other in the retina, and if they're receiving information from the same area of visual space, we can have higher visual acuity for detecting differences in light intensity, color in an object, a scene, depth in a person's face, and so on. So I would encourage you, as you are reading in your textbook, to read the research done by Hubel and Weisel in your textbook. They found that cells in the visual system have what are called receptive fields. Basically, not all of our cells are going to respond the same way to light changes in the same part of visual space. So a receptive field is basically the portion of visual space that will elicit a response in a neuron. Some cells become more responsive when light is shown in the center of its receptive field, and some will actually become more excited when light is shown outside of the center in the receptive field. Now, Hubel and Weisel largely looked at this with cells in the primary visual cortex, which we'll talk about later. But this doesn't just happen in the cortex. It actually begins in our retinal layers. So what you're looking at here are images. Well, they're not images. These are theoretical. These are receptive fields for ganglion cells. So receptive fields are actually going to be formed by the different bipolar cells that are connected to it. So if I shine So here you can kind of see if I shine a light in the center of this on cell, I get more responding in the cell. I get more glutamate release. In the case of ganglion cells, I might get more action potentials. But if I shine light in an off area, that actually has the effect of inhibiting the cell. You actually see less firing than at rest. And we see the reverse with an off cell. I actually get inhibition when I shine light in the center, and I get greater firing when I shine light in what is called the surround. Now again, why do we have these fields? As I have mentioned, this is the first step in noticing differences in contrast. You need to be, if you want to be able to identify objects or see objects in your visual environment, you have to be able to notice the contours and the edges of objects. And oftentimes, contours and edges of objects and being able to separate an object from a background is going to be by noticing changes in contrast, shifting from dark to light. Now, let's take the two pathways that we just talked about, those on and off cells, and look a little bit closer at their receptive field. So if we're talking about a ganglion cell, inputs to a ganglion cell are going to come from nearby places on the retina. So the receptive field of a ganglion cell is going to be the area of space that that cell monitors. Now, both the receptive fields of bipolar cells and ganglion cells have two parts. They have a circular part in the center and then the circular part that surrounds that, which we are calling the surround. These cells, basically, whether on or off, respond best when we have differential illumination to the center versus surround. So in other words, the firing of ganglion cells, and in particular, we're talking about ganglion cells because they fire action potentials, bipolar cells do not. Ganglion cells are going to fire more often when the light is not even across the receptive field. So if I shined a diffuse light that covered both the center and the surround of a receptive field, the center and surround would cancel each other out. So we want there to be differential light. So for example, here we're getting more light than in the surround. As a result, we get a greater change. Here, we get more light in the surround in the center, and again, we get a bigger change compared to rest. So the firing of ganglion cells is going to be greatest when the light is not even across the receptive field. So as a result, these cells are going to be sensitive to contours and contrasts. So let's start by looking at if it's firing action potentials. We're going to go ahead and call this a ganglion cell. So the on bipolar cell has the same center surround pattern as an on ganglion cell, except bipolar cells don't fire action potentials. On cells are depolarized when light hits the center of their receptive field. They're going to fire more action potentials. When light hits the surround, they're going to be inhibited. And as I've said, the off cell will basically do the opposite of that. So now, just as kind of a review, so our photoreceptors are going to be hyperpolarized in light. They're going to be depolarized in the dark. And in light, they are going to release less glutamate onto bipolar neurons. We have our on-bipolar neurons, which are hyperpolarized by glutamate. It works with a potassium channel. As a result, potassium leaves the cell and takes all of that positive charge with it. They're depolarized in the light. They are going to release more glutamate onto on-ganglion cells in the light. And as a result, the on-ganglion cells are going to get more action potentials. With off-bipolar neurons, they're depolarized by glutamate. Again, we have a sodium channel. Sodium is going to enter the cell. They are going to become hyperpolarized in light because those sodium channels cannot stay open. They're going to release less glutamate onto off-ganglion neurons in the light. And as a result, those off-ganglion cells are going to fire fewer action potentials. Now, with bipolar cells, we had clear on and off cells, but that's not actually the case with ganglion cells. They all have the same receptors and ion channels that are opened by that glutamate. So whereas on bipolar neurons had those critical potassium channels and off had those sodium channels, on ganglion cells don't have special potassium channels. Off ganglion cells don't have special sodium ones. They look the same, but we just call the ganglion cells on and off based on what type of bipolar cell is connected to them. So I want to next talk about the role of the horizontal cells and of our amacrine cells. So horizontal cells are going to connect bipolar cells to photoreceptors, and then our amacrine cells connect bipolar cells to ganglion cells. So where do the differences in our center surround come from? So let's talk about why and how the receptive fields are set up this way. Now, as I mentioned before, our horizontal and amacrine cells are inhibitory interneurons. They are basically there to connect the different layers of neurons together. So both amacrine cells and horizontal cells release GABA and glycine, both of which are inhibitory neurotransmitters, and those are going to cause the cells to release less glutamate. So when they are excited when horizontal cells are excited by glutamate release from photoreceptors in the dark, they're going to cause those inhibitory neurotransmitters to be released on nearby photoreceptors. So for example, if this cone is depolarized, it's going to excite the horizontal cell, and the horizontal cell is going to send inhibition to neighboring photoreceptors. And that's going to cause those neighboring photoreceptors to release less glutamate. This is going to become really important for creating that center surround structure. So here's the last slide today. So the horizontal and amacrine cells ultimately are going to establish center surround receptive fields that we see in the bipolar cells and the ganglion cells. So let's talk a little bit again about those layers of the retina to help. Again, horizontal and amacrine cells are going to be those inhibitory neurons. So we can see in this particular case, if we have diffuse light shown on all three of these photoreceptors, here's our bipolar cell, here's our ganglion cell, and here are some horizontal cells. So in the case of diffuse light, we get excitation right onto the bipolar cell, but the horizontal cells, so you can kind of think of this photoreceptor as being the center, and these two basically beginning to form the surround. Here, we get light shown on the center photoreceptor, and that has the effect of exciting the bipolar cell. But at the same time, the horizontal cells that are connected to the neighboring photoreceptors in the surround are going to get inhibition. And as a result, we're getting excitation from the center photoreceptor, and effectively, thanks to the horizontal cells, we're getting inhibition from the surround. So diffuse light not only excites the bipolar cell, but the surrounding lateral inhibition is going to basically cancel out that excitement. That's why with diffuse light, we don't really see a change in cell firing, because while we are getting excitement from the center, we're getting inhibition from the surround. Now, let's say that we have some differential light. In this particular case, here we have light only shining in the on portion of our receptive field. That is going to excite the bipolar cell, and because there's no light here in the surround, the horizontal cells don't really have to, they're already inhibiting these photoreceptors, so they're not going to cancel out any of that excitation that is being sent to the bipolar cell. As a result, we get lots of action potentials. Here, if we have light in the surround, the horizontal cells will actually send inhibition. The bipolar cell will be inhibited by the neighbors. Light excites these horizontal cells, which send inhibition to the bipolar cell. As a result, we get fewer to no action potentials. So in this particular case, we are getting what is called lateral inhibition. We are sending inhibition to neighboring areas of a receptive field so that we can get greater contrast. So, folks, this is all I have. We will start on lecture 15 on Friday, and I will see you all then. Bye.